Good afternoon and welcome to the lecture on molecular-based assays for clinical research. My name is Professor Natalie Abai and I'm the academic head of the School of Clinical Medicine Laboratory. Firstly, what is molecular biology? Molecular biology is a field of biology that studies the composition, structure and interaction of various cellular molecules such as DNA, RNA as well as protein. Now, molecular biology spans across various disciplines from genetics all the way to virology. So we ask ourselves the question, why do we need to isolate and purify DNA or RNA? Firstly, DNA is of primary importance for studying the genetic causes of disease, as well as for the development of new diagnostic tests. It is also essential for carrying out forensic science, sequencing of genomes such as the Human Genome Project, and more recent, the sequencing of the COVID-19 genomes in order to identify different variants across different geographical regions. DNA is also used for detecting bacteria and viruses from different environments. There are various sample types that can be used for the extraction of DNA and RNA. The human-based samples include whole blood, hair samples, a variety of different swabs, throat swabs, genital swabs, rectal swabs. Stool can also be used for the extraction of DNA in order to study the gut microbiome. Sputum sample is used for the detection of MTB. DNA can be extracted from this sample. And urine sample can be used to extract DNA to identify various pathogens. On the more microbial side, DNA can be extracted directly from bacteria, fungi, protozoa or parasites, as well as viruses. There are four fundamental steps in the extraction of DNA or RNA. The first includes cell harvest. This is then followed by rupture of the cell wall in order to release the DNA particles, the cellular particles. This is then followed by the purification of DNA and RNA, because when the cell bursts, it's not only the DNA RNA that flows out, it's also other cellular components such as lipids as well as proteins. And finally, concentration of the DNA solution. Look at the first step, which is cell harvest. This is a very important step because it is used to concentrate the cells which contain the DNA. And this is accomplished by using a special instrument called a centrifuge. During the spinning of a sample, be it blood sample or a urine sample or sputum, the cells which contain the DNA clump to the bottom of the tube, as indicated in the little diagram shown on the right hand side. The next step is cell wall rupture and cell extraction preparation. Now, there are two various methods that can be used for this process. It is, not two various methods, there are two common methods that can be used for this process, and this includes the physical method and the chemical method. Now, the physical method basically uses force, and this is accomplished either by vortexing or sonication. The majority of research labs do not have a sonicator, so most of them opt for the chemical method, which uses chelating agents such as EDTA, as well as surfactants or detergents such as SDS, in combination with an enzyme such as lysozyme. So either of these two methods are quite acceptable for the lysis or the breaking of the cell wall. Once the cell wall is broken, the next step is to now try and purify the DNA from the remaining cell components, and this includes proteins mostly. This can be accomplished by organic extraction or enzymatic digestion. Now, the majority of the labs tend to use the more organic extraction procedures, which include a mixture of phenol and chloroform. When phenol and chloroform are added to this DNA extract, the DNA floats to the top after centrifugation, and the middle mass includes all the cellular debris, and the phenol and the chloroform remain at the bottom of the cell. So this is quite a nice way of separating the different components that are present in the cell. Once you have purified your DNA from the cells, the next step is to precipitate it or wash it. And one of the most common chemicals that is used for washing is ethanol. Isopropanol is also acceptable. Although these chemicals wash the DNA, they do not degrade or interfere with the DNA. So the final step is concentration of the DNA. So once the DNA has been concentrated, it is now ready for downstream application, be it sequencing or PCR. Some researchers prefer to visualize the DNA before moving to downstream applications to ensure that the DNA that is extracted is of high quality. For DNA visualization, one of the processes that is used is called electrophoresis, which involves the movement of charged particles in a fluid or gel under the influence of an electric field. Now, this technique can be used to separate both DNA, RNA, as well as proteins. DNA is negatively charged, so therefore it will move to the positive pole. This slide represents a nice diagram of the entire electrophoretic process. DNA is clear after extraction, therefore it won't be able to be viewed once loading into the wells. It is mixed with a sample loading dye, which gives it a purplish color. This dye also gives the DNA density so that it doesn't float when it's under the buffer. It basically sinks to the bottom of this gel. This gel agarose is made from seaweed and it's a nice tightly packed gelish matrix. Once the DNA is loaded on the at the negative pole, it is then subjected to an electric source or a voltage. A buffer is present. The buffer has a good ionic strength to support the movement of the DNA from the negative to the positive. DNA cannot be viewed with the naked eye after it's run on the gel. It requires a UV light, and this is viewed using a UV transilluminator. So under the UV light, DNA fluoresces orange, but when we capture the picture, we see a black background with white lanes which represent the DNA. Researchers are unable to tell the size of the DNA extracted, therefore they run a molecular marker which looks like a ladder. Each little band has a specific size, DNA size, so from this band we can tell the size of the DNA. Also, researchers like to look at the integrity of the extraction. If you look at the bottom of the slide, the smear that is shown is basically a bad extraction because the DNA has been degraded or sheared into tiny little pieces, therefore it looks like a smear. However, DNA that is intact will look like lovely, clear and crisp bands on the agarose gel. So let's move on to the crux of this presentation, which is molecular methods and clinical research. So the emergence and re-emergence of several infectious diseases can potentially impact public health worldwide. And this has become a topic of particular interest in clinical research. Molecular methods can complement conventional culture, antigen and antibody-based methods for the detection, identification, and epidemiological analysis of infectious microorganisms. However, the disadvantages of the conventional methods, which are the culture and antigen and antibody-based methods, is that they are unable to distinguish new pathogens derived from a common ancestor. They're also quite time-consuming, and sometimes the results that they provide need to be confirmed by other tests, so they're not that accurate. Molecular methods, therefore, are now emerging as important tools in all laboratories for routine detection and fingerprinting, as well as aiding in public health surveillance, which could potentially allow the rapid implementation of infection control and intervention practices. One of the most powerful molecular diagnostic techniques used in the clinical lab is the polymerase chain reaction. You have already come across this technique, known as PCR, which amplifies millions of copies of a target DNA from as little as 10 picograms of DNA. PCR has enabled researchers to identify the causative agent that has been associated with a variety of different diseases, with a detection limit that varies from 1 to 100 copies of a target amplicon, which is far superior to its equivalent immunological assays. So even a low concentration of DNA, and even DNA that is not of a very high quality or purity, can start amplification reactions. Now, apart from the conventional PCR, which involves the running of an agarose gel to view your target gene, real-time PCR has further increased the accuracy of the normal PCR. Real-time PCR assays use selective primers and probes which target specific genetic sequences. They are more sensitive than the conventional PCR. Apart from its high sensitivity, real-time PCR is also less time-consuming. As the amplification is taking place, similar to what its name suggests, you can view the results in real time. You do not have to wait for the reaction to complete after 2 hours to then run a gel for a further hour to determine if the sample or the target gene is present or not. Real-time PCR also does not include the use of carcinogenic agents such as ethylium bromide, which has been used for the staining of DNA gels historically. A decrease in turnaround time through molecular testing is another advantage using over conventional culture-based testing. With a real-time PCR assay, you can get a result for a pathogen in about 46 minutes, whereas with a conventional culture-based assay, it may take up to 5 days to get the same result. There are different variations with techniques associated with real-time PCR. You get the more traditional type of PCR where you extract the DNA first and then use the specific primers and probes that target that pathogen that you are detecting. The technology has become more sophisticated in that now we have closed systems. You would have probably heard of the most famous system known as the GeneXpert, which is a point-of-care system for the detection of rifampicin-resistant tuberculosis. In this type of real-time PCR system, there is no need for the first step of DNA extraction. The sputum sample is simply loaded into the machine, DNA is extracted, and amplification takes place thereafter on the extracted DNA. So this type of system has even increased the efficiency of the conventional traditional real-time PCR, and it is a platform that is most commonly used now in diagnostic laboratories because these instruments are approved for diagnostics. They have FDA approval. So let's look at a scenario. We did say that molecular-based assays are more superior to conventional culture-based assays. So let's look at how this is possible. So a young female visits the STI clinic in Baltimore, USA, with symptoms of greenish yellowish discharge and lower abdominal or pelvic pain. The suspected pathogen to the clinician is Neisseria gonorrhea. In the United States, they do not follow syndromic management of treatment. They would have collected the sample and they would have diagnosed it before treatment is initiated. So let's compare the diagnostic methods of classical culture versus molecular detection. With classical culture, firstly, the young female would have to provide an endocervical swab, which is a clinician-based sample. The swab would then be plated onto selective media and incubated for 24 to 48 hours. Let's look at 48 hours being the optimum for the growth of any pathogens on the plate. After 48 hours, a few presumptive tests will need to be done, or confirmatory tests, including microscopy and biochemical analysis. At this stage, there is still no further confirmation. Suspected pure isolates will then have to be subcultured and further incubated for about 24 to 48 hours. After this, it will go through the same route of biochemical assays, and lastly, a nucleic acid amplification test can also be used to confirm identity. All in all, this could take up to 5 days just to say yes or no, is it Neisseria gonorrhea? Look at a molecular detection assay, such as the fluorotype Neisseria gonorrhea assay. So with this assay, what we simply do is, firstly, we take a urine sample, urine versus endocervical. The urine sample the female can collect herself, and it's less invasive. She doesn't need to see a doctor and undergo a pelvic examination. The urine sample is placed directly into a vial in the machine that is specific for this microorganism. The DNA extraction takes place and amplification takes place. The result is available in approximately 3 hours. Now let's look at it. 5 days as opposed to 3 hours to diagnose this pathogen. With the molecular assay, treatment can be initiated within 24 hours, whereas with the culture assay, treatment can only be initiated after 5 to 7 days. So the advantage of molecular assays over classical culture, which I've already mentioned: faster turnaround time for results, can detect multiple pathogens from a single sample, there's less sample handling, and also non-invasive sample types can be used for detection, urine versus endocervical swabs. So molecular detection of various pathogens. Let's go on to respiratory pathogens. Clinical laboratories historically diagnose 7 or 8 respiratory virus infections using a combination of techniques which include ELISAs, antibody staining assays, cell culture, and also molecular-based assays such as nucleic acid amplification tests, with the most common being the PCR. With the discovery of 6 new respiratory viruses since the year 2000, laboratories are now faced with the challenge of detecting up to 19 different pathogens from a single sample, which is associated with upper and lower respiratory tract infections. Now, 7 or 8 versus 19, this number has actually doubled. So the application of nucleic acid amplification technology, particularly multiplex PCR, provides an important tool. So what is multiplex PCR? Multiplex PCR means that you can detect up to 6 or 7 or even sometimes 10 pathogens from a single sample using this type of approach. So let's look at the panel that is highlighted in the red block. So these are specific assays, real-time PCR assays that can be used for the detection of various respiratory pathogens. So from a single nasopharyngeal swab, you can detect all the viruses that are shown in this little panel, viruses and bacteria that are present in this little panel. So it has actually revolutionized the way diagnostics is currently being done. So let's look at this research paper that was published a while back. So this study looked at surveillance data on Mycoplasma pneumoniae infections in children in Beijing. So children who were diagnosed with pneumonia or respiratory tract infections were part of the study. Samples were collected from January to December 2015, so it's just done in a single year. And the samples included throat swabs, bronchoalveolar lavage fluid, as well as sputum. So from these different sample types, DNA was extracted and was used to detect Mycoplasma pneumoniae by real-time PCR. Now remember, this was a surveillance study, so every single month they were looking to detect this pathogen and determine the frequency of this pathogen. So based on the study that was done over the course of this 1 year, they found that the epidemiological patterns of M. pneumoniae were increasing. So they weren't declining as the months went on, they were actually increasing as the months went on. And this indicated to the researchers that the epidemic outbreak may continue into the following year, which was 2016, and there was no way that the epidemic was going to end in 2015. So this was a nice way, using a DNA-based assay and real-time PCR, to track the spread of and the frequency of infections in that specific population. So let's go on to different pathogens such as gastrointestinal infections. So molecular-based diagnostic methods have been used for the detection of gastrointestinal pathogens, and these methods are becoming more popular in microbiology laboratories. So again, multiplex PCR has shown to be superior when compared to culture-based assays, since they have shorter turnaround time and you can detect multiple pathogens. So let's look at the panel that is shown here. So from a simple stool sample, you can perform a real-time PCR, a multiplex assay, to detect all of these microorganisms that are present here that are associated with gastrointestinal infections. So from a single sample, you are able to tell what is the frequency of each of these different pathogens. So let's look at a paper that was published that is associated with molecular detection of a gastrointestinal pathogen. So let's look at Campylobacter jejuni in patients with Crohn's disease in Iran. So for this study, patients diagnosed with inflammatory bowel disease, as well as healthy individuals with the controls, were included in the study. So biopsy samples were obtained from these patients. DNA was extracted from the biopsies, and it was used for PCR and real-time. It was used in PCR and real-time PCR assays. So normal PCR and real-time PCR assays. So this is the normal PCR. The real-time PCR, you won't be needing to run a gel, so you won't see this. So what is the take-home message from the study? The research findings demonstrate that Campylobacter jejuni may pose a predisposing factor in the development of inflammatory bowel disease. And more research is needed to detect the cause of inflammatory bowel disease and identify the correlation of CGHNI with Crohn's disease in Iran. So using this simple molecular-based assay, you are able to draw correlations between infection and pathogen and causative pathogen. So, rapid molecular detection of gastrointestinal pathogens and its role in antimicrobial stewardship. So for this study, they looked at Clostridium difficile in children and Salmonella species as well. So patients were diagnosed with acute diarrhea. Stool samples were also collected, and they performed real-time PCR for 20 pathogens. They compared the real-time PCR to stool microscopy and culture to show the sensitivity and specificity of the molecular-based assay as opposed to the conventional classical microbiology. And what did they find with the study? There were high detection rates of Clostridium difficile in children and Salmonella species, as well as relatively high rates of Campylobacter species, which were hard to isolate by routine culture. So therefore, the findings of this study show that non-culturable isolates are no longer an issue for detection because molecular-based assays can even pick up the non-culturables. So let's look at infections associated and applications of PCR-based assays in central nervous system infections. So the increasing availability and use of molecular tests for the detection of microorganisms from CSF has redefined our approach to common CNS infections such as meningitis, encephalitis, and brain mass lesions, particularly in patients who are immunocompromised, such as those that are HIV infected. So the use of molecular biology has also improved the ability to identify the etiological agent responsible for other central nervous system syndromes, such as transverse myelitis. So if we look at the little table that is present on the slide on the right-hand side, by using a CSF sample, a multiplex real-time PCR can be performed to identify all the microorganisms that are associated with central nervous infections. Keeping it with the theme of central nervous infections, specific viral pathogens can also be identified that can be associated with these infections. And these viral pathogens include human herpes viruses 1, 2, 3, and 6. Now, using a CSF sample, if you have already available in your lab for studies that have been previously conducted, DNA can be extracted from the CSF. However, the sample population that needs to be targeted are patients with acute meningitis or encephalitis. DNA can be extracted using a viral DNA extraction kit because we're working with viruses, which is human herpes viruses. We're no longer working with E. coli, which is bacteria. And real-time PCR can be performed to identify these 4 viruses. And this was done in the study that was published in 2018. And this study found that the most frequently detected virus by PCR was herpes simplex virus type 2, followed by HSV1. And in the case of encephalitis, the most frequently detected virus was HSV1, while HSV2 was the most frequent in case of aseptic meningitis. So this is a nice way to also link a clinical syndrome to that of a causative pathogen. So let's look at sexually transmitted infections. So the Thermo Fisher has now put out or has made commercially available primers and probes for the detection of various STIs by the real-time PCR. And this allows for the rapid identification of these pathogens. Within 46 minutes, you can identify the causative agent of the syndrome that you are trying to investigate, be it vaginal discharge syndrome or cervicitis. So these probes and primers have worked remarkable in a number of research labs, and there are various publications from them. If you want to look at the more conventional approach where you perform a PCR and run a gel, this paper was published by a Brazilian group where they were able to detect several STIs using a multiplex PCR approach as shown in the table and running a normal gel. And each band on this gel represents the size corresponding to a various STI. So what the researchers found in the study is that this method enabled the detection of 7 STIs. And the advantage of using the multiplex PCR is that it simplifies the workflow, enabling the performance of these assays in routine diagnostic laboratories with basic molecular biology facilities. When we say basic molecular biology facilities, real-time PCR instruments are expensive. They can cost anywhere from 500,000 upwards, whereas conventional PCR machines cost about 30,000. So these researchers showed that even with low-cost instruments, you are still able to get good quality results. So let's look at more of the non-communicable diseases now. Let's look at breast cancer. So over recent years, progress in molecular techniques has made it possible to analyze FFPE tumor material from larger cohorts of patients. This has incentivized many translational studies relating to molecular tumor markers for diagnosis, prognosis, and response to therapy. And they have resulted in many relevant molecular markers that have rather quickly made it into clinical pathology practice. So basically what they're saying is, using these stored tissues, you were able to detect specific genes that are associated with disease progression or specific genes that predispose women to breast cancer. So this table provides an overview of the various molecular tests for breast cancer and their current clinical utility. So this paper was published by a group from the Netherlands. We would see that using real-time PCR, and you were able to detect primary tumors. Also using fluorescence in situ hybridization, you were also able to detect adenoid cystic carcinoma. Using microarray assays, which are more sophisticated as opposed to the normal real-time PCR assays, you are able to look at patients. You are able to look at various cancers as well, breast cancer, and sequencing, which is also a really hot topic right now, especially whole genome sequencing. Using just one single sample, you are able to understand every single gene that could be associated with disease, predisposing you to disease, as well as progression of the disease. Then let's look at diabetes. So type 2 diabetes is the most often considered as maternally inherited disease, which involves a mutation from adenine to guanine at position 3243 on mitochondrial DNA in a specific transfer RNA gene. Now, this study looked at the molecular genetics of type 2 diabetes associated with this mutation, the adenine to guanidine, in mitochondrial DNA in a Pakistani family. In this study, so they brought in the mom, dad, and the kids, and a blood sample was extracted from this family. They did a PCR to detect this gene, this transfer RNA leucine gene. After they had detected it, they sent the sample for the PCR products for sequencing. And when they sent it for sequencing, the idea was to try and identify this adenine to guanidine mutation. They found after sequencing that this mutation was not found to be frequent cause of diabetes in the study population. And this finding suggested that there might be some underlying genetic cause for type 2 diabetes mellitus in the subjects there may be some other underlying cause for type 2 diabetes mellitus in the subjects under study, because there was a deviation from the classical known mutation. So let's look at assays for congenital heart disease. So molecular diagnostics has been integrated into routine clinical healthcare practice for patients with congenital heart disease. So if you look at this really nice diagram, it shows a molecular test that can be used. For example, for trisomy, you can use karyotyping. If you want to look at DiGeorge syndrome, there's fluorescence in situ hybridization. And for other symptoms such as congenital heart disease and familial congenital heart disease, you can use targeted sequencing. With fluorescence in situ hybridization, you would require using fluorescent probes. The process does take a long time because they have various hybridization steps, and sometimes these steps need to be confirmed by whole exome sequencing or whole genome sequencing, so there are further steps. With the targeted sequencing, if the researcher knows the specific gene that carries the mutation that is responsible for causing the disease, that specific gene is amplified by normal PCR. It is then sent for Sanger sequencing, and mutations can be detected, as shown in that the study with type 2 diabetes. They would look at to see if the specific mutations are available. So FISH, which is fluorescence in situ hybridization or karyotype analysis, is suitable for genetic testing of patients of suspected Down syndrome, while Sanger sequencing is perfectly applied for those with suspected Marfan syndrome. And that is where we end our lecture for today. I thank you for your attention.